All right. Hi, everyone. My name is Dr. Eryn Slankster-Schmierer. Any three of those names is fine by me. And I'm going to tell you today about my journey from working in academia as someone who experimented on animals to working in Animal Protections Agency. So I started and ended my university education at Oakland University in Rochester, Michigan. And here I started my education with a bachelor's degree in molecular biology. After my bachelor's degree, I felt a little more, I needed a little more guidance. So I went on to get a master's degree, driven largely by not wanting to experiment with animals. My thesis focused on creating genetic markers in a plant for use in a teaching laboratory. And I knew before I even finished my master's that I wanted to get a PhD. So during my master's studies, our university hired an enthusiastic professor. He was studying stem cell and cancer biology. His research was super exciting to me, and he was recruiting grad students. I never actually wanted to perform animal experiments, but I was desperate and I was desperately seeking work that I thought would translate to human health outcomes. So somehow I convinced myself that I had to work with animals to do this. And long story short, I was sold with the hope of finding solutions to a problem that three of my grandparents were actively losing their battle with. And so for the next several years, in collaboration with a biotech company, I studied the role of the immune system following surgical induction of critical limb ischemia in a mouse model. This is a highly common disease associated with heart disease, diabetes, and atherosclerosis. I simultaneously studied the mechanism of an autologous multicellular stem cell therapy to treat it. I used mice as an experimental model for those 5 years, and I could go on and on about how this created cognitive dissonance with me. I was studying a disease in a species of animal that never naturally got this disease that we were studying. We often performed experiments in mice without immune systems at all, trying to mimic a highly immune-dependent human-specific disorder. I struggled trying to justify experiments that failed to mimic human disease over and over again. And I left most of the ethical conflict out of my presentation that brought me here, but please feel free to reach out to me if you're interested in talking about that at all. But as I was finishing my PhD, I knew that I couldn't work with mammals anymore and I needed a career shift. And so from there, I took a hard left in order to avoid mammal work, and I found myself looking at fruit fly labs. I brought my molecular biology experience to a behavioral neurobiology lab at the University of Nevada, Reno. Here I studied the molecular mechanisms of olfactory response, or the sense of smell, in a fruit fly. I studied the effects of diet and gut microbiota on olfactory behavior of fruit fly larvae. I studied their effects on gene expression within the olfactory neurons and how genetic manipulation of these genes affected the adult feeding behavior. I really enjoyed the scientific process. I published a couple papers, but I was becoming sort of disenchanted with the academic options that I saw before me. Again, there's nothing wrong with that option. I just was learning they weren't for me. And long story short, my supporting grant funding for my postdoc ran out during COVID-19, and for the first time in my adult life, I was unemployed. For me, this was a blessing in disguise. I wish I could have seen that this was like a pivotal moment back then of opportunity in my life, but that's not how it went. But I began seeking employment options that aligned with my ethics. So that's a good thing that came out of this. And well, I don't think you should wait for unemployment to seek new options. This pushed me personally to reassess my priorities and to think about the types of work that would make me happy. I was forced to confront that I did not want to continue the academic route. Based on my experiences with industry in my PhD, I was pretty sure industry wasn't for me either. Again, nothing wrong with it. It's just not for me. It doesn't suit my personality. And I knew that I didn't want to experiment on animals of any kind anymore. And for me, I kept finding myself looking at nonprofit organizations. So for a while, I struggled to find the value in my experiences. I couldn't help but think that my history seemed really scattered and not convincing enough to get the type of jobs that I wanted. But ultimately, I ended up gaining a lot of experience in a broad knowledge base. And this allows me to relate to a lot of different job postings, not a few. And so it's important to remember that, especially as an early career scientist, scientists are trained to solve problems and they're trained to ask the right questions. While going to grad school, we aren't necessarily signing a contract for a specific specialty, and there's usually an ability to change direction. And I cannot stress this enough. Your best skills may not be in the title of your dissertation. And this was a huge lesson that I learned. Which brings me to today. So for 3 years now, I've been working with the Physicians Committee for Responsible Medicine. We are a mission-driven organization that is dedicated to saving and improving human and animal lives through ethical and effective scientific research. We are an NGO, which is a non-governmental, nonprofit organization, and we are funded by private foundations and individuals who support our mission. So at the Physicians Committee, the research and regulatory team works to replace animal use in science and medicine. Finally, I found a team that was showing that we can advance science and human health effectively without the use of animals. I do this by advocating for more effective, human-relevant, non-animal methods in regulatory toxicology. And one of the ways I do this is by developing a training program called NEURA. So NEURA stands for NAM Use for Regulatory Application, and through it, I provide trainings on non-animal methods aimed to replace existing animal methods. I regularly work with experts from academia, industry, EPA, FDA, as well as global regulatory bodies as well, and regulators. So in this way, I provide non-animal education to those who seek it, and I facilitate conversations that help overcome regulatory challenges in accepting non-animal methods. So I also lead the International Council of Animal Protections within the OECD, also known as ICAPO. So the OECD is an international organization that works to create harmonized policies for its 38 member countries, including the United States and Europe. These member countries, by association, agree to the guidelines outlined within the OECD. So, by having a seat at the OECD table, this allows us a tremendous opportunity to influence guidelines and to work towards international replacement of current animal testing requirements with more predictive, human-relevant, non-animal approaches to safety assessment. So, finding a job within an NGO like the Physicians Committee, and there's certainly lots others, offers a really unique opportunity. When you work for a nonprofit, you're working towards a cause that you believe in. And at the Physicians Committee, I represent members who want to drive change, and I provide the expertise to facilitate this vision. So this affords you the opportunity to work in ways not always available to academia, industry, government, etc. As an academic, for example, it may be difficult to get funding for the type of work that we do. As an industry scientist, you're mostly constrained to that which benefits your organization's specific needs, not necessarily your curiosities. And as a government body, you could be constrained to representing your constituents as a whole and not necessarily your own personal values. Of course, this really applies to all sectors, but it's about choosing which constraints that you prefer to live with. So NGOs are often responsible for communicating between public and various sectors. We have the luxury of advocating our members' collective mission across these sectors, and in return, we keep our members engaged on important news that concerns them. We also have the ability to influence and shape policy, regulation, and company decisions. We can do this by holding agencies accountable for promised changes, by tracking their progress. We can provide ideas and in-kind labor to various projects to facilitate their progress, such as my work with ICAPO. Or we can provide education through training or lobbying, like I do with my neuro work. And of course, sometimes we can even provide funding for projects to facilitate our mission. So working at an NGO also offers a unique opportunity to develop relationships across all sectors, and this is probably my favorite part. My work with NEURA and ICAPO are great illustrations of the importance of collaboration. We have the opportunity to bridge gaps among each of the sectors. And we can do this by facilitating conversations across the sectors or perceived boundaries, and allowing us to build off of each other's experiences and find common ground that allows us to progress mutually. Without NGOs facilitating these conversations, sometimes they simply may not be able to happen. They may not have the opportunity to happen. And so finally, as an NGO, we are able to support other scientists by promoting their work, which is also, at least for me, really fun. So as a scientist who started working with animals and ended up advocating against that, I really want to emphasize that you have more options than you think. And so whichever sector suits you the best, you certainly don't have to work with animals to make an impact, which of course ITSE did a fabulous job demonstrating today. And so will the upcoming presentations from the GRC and Hasbros. With that, I just wanted to thank the organizers for allowing me to give my career perspective today. And if anyone has any questions at all, my email is at the bottom of the screen. You are welcome to contact me with questions about literally anything, about my work or how I got here or anything else. Thanks, everyone. That's it.
Thanks, Dr. Slankster-Schmierer. Next, we'll welcome Dr. Gastaldello.
Hello, hi, everybody. So I haven't prepared actually a presentation about my career, so I just put a slide showing where I'm working now, which is, and then I give you a bit of my background. So at the moment I work at the European Reference Laboratory for Alternatives to Animal Testing, which is part of the Joint Research Centers, a research center of the European Commission. We are based in Italy, and as a job, within the center, within ECVAM, as the European Reference Laboratory for Alternatives is known, we do research and development of alternative methods. We validate them, we disseminate the 3 Rs, the concept of the 3 Rs, so replacement, reduction, and refinement, and we promote non-animal approaches in science. So I started as a researcher, like probably a lot of my colleagues. So I have a degree, a master degree, a bachelor and master degree in biotechnology, medical biotechnology, which I got in Italy. After that, I had the opportunity to go to the UK, to London, to do a bit of a research project, and then I stayed there for 13 years. So what I did there was find a PhD, which was done in Edinburgh, in cardiovascular research. And that's, well, even before my PhD, I was working with animals, actually. I was working with rats in London. I wasn't comfortable about it, but I think I wasn't that uncomfortable either. So my PhD was still using animals, and that's when I started getting uncomfortable in using animals. So my PhD was in cardiovascular science, but I was using a rodent model of dermatitis, skin dermatitis. And I was doing some in vitro work, but always using animal cells and animal-derived material. So when I started, having to kill animals, actually, I started to get uncomfortable. And at that time, I knew I didn't want to continue doing that. I was still uncertain whether to get out of academia or not. So I just kept going in academia, and I looked for a postdoc after my PhD. I have to say, at the same time, while I was doing my PhD, I got to know of the existence of ECVAM, so the Center for Alternatives to Animal Testing in Europe. But my impression was that it was very difficult to get in. It was just an impression, probably more influenced by my own insecurities and my more uncertainties of what I wanted to do with my career and my life than, reality. But I got to know the existence, and I guess it stayed at the back of my mind. But I didn't try to apply for a job there. I just looked for a postdoc which didn't involve working on animals. I ended up working for animals, meaning I got a postdoc studying a transmissible cancer in Tasmanian devils, which Tasmanian devils, if you know, they are marsupials present only in Tasmania, which is an island, an Australian island. They are affected by this transmissible cancer, and the species was at least at risk of extinction. So I found a job actually which was in the lab using animal materials, because I was using animals-derived cells. But of course, the material was used for the purpose of saving this animal or saving this species. After my postdoc, this was still in UK. I was a bit tired of the academic environment, I have to say. I didn't see myself becoming a PI, a principal investigator. I didn't see myself fighting continuously for funding and having the pressure of having to publish constantly. I also wanted to go back actually to Italy. So, sometimes our careers and our path are defined by more than one factor. It's not just about what you want to do as a career, it's also what you want to do in your personal life. So I moved back to Italy without a clear plan. I started actually doing a master in nutrition from UK, and then I finished it when I was in Italy. I spent 2 years in Italy doing different things. I was actually, for a year, I was growing and selling organic vegetables together with my sister. We were planning actually to open a cafe. I was doing COVID testing because I went back when COVID was ending, let's say it was 2021. So I was, with my degree, I could do COVID testing. So I was working also with that. So I had a bit of a couple of years of trying to different things. I didn't know exactly where I was going. One day, during my master in nutrition, I was reading a paper published by my supervisor of the master and by another woman. And for some reason, I decided to check what this other woman writing the paper with my supervisor was doing at the time. Probably to kind of understand, because she actually was working with my supervisor in nutrition, I thought maybe I can understand where she is now, what she's doing, I can get some inspiration. So I actually went to see, I checked her name, I went to LinkedIn, and I found out that she actually used to work for the European Commission for ECVAM, so where I'm working now. And that kind of reminded me of the center, ECVAM Center. And it happened that at the time they had a position open which fit my profile. So I also reached out to this girl to kind of ask for information about the center. And I applied for the job and I got the job. So this was in 2022. So I started with this job at the European Commission in September 2022 as what is called a contract agent. So this is not a permanent position, it's a 3 years plus 3 years contract. Just to a bit of technicalities in case somebody is, interested, and it's not easy to get a job as a permanent staff at the European Commission, but contract agent is a way to start and to get to know the place and how it works. And so since September 2022, I've been working on alternatives to animal testing from, let's say, in the biomedical research sectors, which is the sector using the most amount of animals in Europe and in the world. And is a sector where I think we still don't talk much about replacement and alternatives because it's a bit more complicated to replace animal studies in basic research or translational research. So my job is very varied, and So I started by analyzing the impact of EU-funded research on society to see if we are funding the right kind of research. Now I'm doing an analysis on animal-based studies approved in Europe, and to see if we can actually replace some of the models used with non-animal approaches, and how best we can do that. Also, I am very interested in education, so I'm working on a few initiatives to engage with students at university levels, but we have also initiative to engage with students at high school level on how to promote use kind of the students and their enthusiasm and their kind of open-mindedness to promote the 3Rs and non-animal method in the education sectors. So it's a very, very job. You start maybe with a project, but you then for sure you're going to get engaged in other projects. It requires a lot of maybe mediation skills, diplomacy, a bit of politics. Is very interesting because it's not just you will do science, but it's not lab science. It's not just science research. Is engaging with different stakeholders, which can be NGOs, which can be funders, which can be journal editors, all those players which you normally find in biomedical research, in my case. But of course, our center is also very much interested and working in regulatory science, in safety assessment. So if you are more interested in that, if your experience is more suitable for that, is an interesting place as well. But the common thing in the unit where I work is this really varied kind of connection that you will get, with these different players around Europe, which you don't normally find if you are just a lab researcher. It's very exciting, eye-opening, but you will need a bit to retrain the way you think about doing research when you do research at ECVAM, because as I said, it's more of a political environment, regulatory environment, diplomatic environment. So yeah, if you are interested in knowing more, please get in touch. I would say I wasn't planning to end up at the European Commission. I'm happy I did. I had a bit of a hard time, I think, getting used to the way it works because of my personality, because I had a past as a lab researcher. But is I guess it's with any kind of job, you will have a period where you need to get used to the new job, how it works, and that's depend on the person as well, how long it will take. So I ended up a bit by chance, but I think that's not a bad thing. And my varied experience, a bit like the person who was speaking before me, I have a very wide experience. Even when I was working in research, I did different topics, and I did different things after leaving UK and leaving research for a small period, and that gave me really a lot of skills, a lot of knowledge that now I feel is useful in this kind of environment at European Commission level, where you have to talk with different people and do different things. So yeah, I don't think you need necessarily to have a clear idea of where your career is going. It might be scary, it might be unsettling, but I think what needs to be clear is probably your principle, your ethics, as the previous person was saying, if you are clear in which realms of, work you want to be a bit, which principle you want to follow, sooner or later, I think you will get there. Maybe go through paths which you hadn't anticipated. But the important thing is you get there, and I think you will get there as long as you have your principles clear in your mind, which for me was, I wanted to stop doing animal research. So that's it for me. I hope it was clear even without a presentation.
Thank you, Dr. Gastaldello. Next, we'll welcome Dr. Hickman.
Everybody, I know I didn't test these slides, but I just have like 4 I want to show. Can you see that?
We can see the slides now, yes.
Yeah, so currently I have 3 jobs. I'm a professor at University of Central Florida, so I've been at for 20 years. Got a group of about 20 people. And then I'm also president-elect of the MPS Society. A lot of you know, we just had our annual meeting in Brussels, it went very, very well. But I'm also here talking about, from the industry standpoint, for Hesperos. So let me kind of just give you a little idea of what Hesperos does, and then I'll kind of go through my career track because it's been kind of interesting. So Hesperos was started about 10 years ago with Mike Schuler, considered the founder of the body and chip field. We've licensed a bunch of patents for the company. Mike and I had won the Lush Prize previous for alternative animal testing. We use a lot of SBIR grants and not a lot of investor funding initially to get the company going. We work with folks all over the world. We got a 14,000 square foot facility here in Orlando, Florida. The company's now up to 50 people. We're a CRO. We do not sell products at all. But I want to emphasize we're the first microphysiological system where our efficacy data was the only data needed for a successful IND for repurposing a drug for Sanofi. And that was successful, and it's now in a 2-arm phase 3 trial, which started in May. So I started off with this idea of clinically relevant functional readouts back in the 90s. And what is that idea is, you go into a doctor's office, he's immediately stick a needle in your arm to be able to pull out fluids, look at biomarkers, right? He looks how you're walking, how you're talking, are you making sense? He listens to your heart. He checks your reflexes. He might ask you about elimination. What he's really checking on is muscle contraction, electrical activity of neurons and cardiac cells, neuromuscular junction function, whether or not your barrier integrity of your GI tract and kidney is working well. So what I've done is to work with taking silicon devices and integrate them with cells, and let's say it would be cardiac cells. And then, so when the cells contract, it bends the cantilever, deflects the laser, I can get force. I can pattern cardiomyocytes over top of microelectrodes to be able to conduct velocity QT interval. I can also see force with the skeletal muscle. We have a wide variety of different neuronal systems, both PNS and CNS, that we're using. We can still look at biomarkers, we don't have to. This allows us non-invasively look for acute, more importantly, chronic monitoring of these systems. But again, without cell death, that's where the clinically relevant comes into, where we have a one-to-one correspondence with these readouts with what a clinician would be expecting from a patient. And then what we can do, we can actually take the cells or the chips, culture them in their own conditions, and to time it such that we can get the maturity at the same time, put them in the base platform, put a gasket on top of it to have a connected microfluidic channel. Then we use, everything we do is in serum-free media. I banned the use of serum in my lab 25 years ago. So everything is all serum-free. Currently, everything is all human. It didn't start out that way because there wasn't human cells available back in the 90s when I first started doing this. But as soon as the human cells become available, we switched over to that. And we can put a clear top on it to be able to do all the advanced imaging studies. And again, say if I wanted to look at pancreatic organoids in the model, I could take out this chip and put another. So it's very reconfigurable as well. And a lot has been made about the recent announcements by the FDA and NIH that are going to move away from animal models into NAMs. One of the things that have enabled that is we've been able to actually get a multiple number of drugs into phase 2 clinical trials using only efficacy data for the IND applications. We've done this for ALS with Apellis, argenx, myasthenia gravis for dianthus, multifocal motor neuropathy for dianthus, CIDP for dianthus, and then I mentioned also the CIDP for Sanofi again, which is now in the phase 2, the 2-arm phase 3 trial. And so basically, that's kind of Hesperos. This is kind of where I've gotten. Now, I'm going to stop sharing and tell you how I got here. So I got my PhD in surface chemistry, actually, which is why the integration of the silicon into the cells, which is why I was very competent at that from very early on. And so my PhD, I worked on some of the foundational aspects of nanoscience, basically, putting on cells onto microelectrodes and not cells, but modulators of microelectrodes, integrate them, creating functional sensors, and also the first time being able to analyze them using surface chemistry or surface analysis techniques. So when I got out, I then went to work for a contract research company, went industry first, set up one of the first bioelectronics labs with collaborators from NRL and from Stanford, and then started applying the surface chemistry to marry up the biological and non-biological aspects of this. And this was a little difficult because I had never taken a biology course at all in my entire career, undergrad, master's, and PhD. So I had to learn cell biology, I had to learn neuroscience. And then we're starting integrating neurons with microelectroarrays that we're looking at them, using them as biosensors for chemical warfare agent detection. And then we started branching out into how to look at these in terms of understanding neuronal communication. So I had to start writing grants into information technology, which we were successful on. And another key thing for the career is, even though I started off in industry, I was publishing the entire time. Now, I then took a job at DARPA, which is Defense Advanced Research Projects Agency, in the Information Technology Office, and started again applying what I've learned into these new areas. A lot of work in terms of brain-machine interface work was first funded in the late 90s out of that program. Some of the work in terms of neuromorphic computing was also funded out of that, some of that work. But then I was offered my first academic job, which was at Clemson University as a endowed chair in a bioengineering department, and as a full professor. And again, I'd never taken an engineering course, so I had to be self-taught in engineering now as well to be able to teach it. And then I basically was successful there, but then UCF came and made an offer I couldn't refuse. And so I then transferred and set up a nanoscience center at UCF, but continued my research in this area and expanding it all along. I invented bioprinting with Tom Boland at Clemson in the early 2000s, which again has spawned, numerous companies and numerous efforts as well. We still use that, but I always consider that as a tool as opposed to something to build a research career around. It just was a means to the ends of trying to integrate cells with silicon-based devices to make these functional readouts. And then founded a company, and so now I've got 3 jobs. And that's kind of how I got here. But it just shows you can learn, just about anything. I've taught neuropharmacology courses without ever taking a biology course or a neuroscience course. I have taught engineering without ever taking an engineering course. So don't limit yourself. Don't say, well, I've only ever done this, I can't do that. Give it a shot. You might be able to do it. One of the ways I was able to do this is I would get well-versed enough in a particular field I was trying to get into, to the point where I knew enough to be able to talk to the smartest people in that field. But without knowing all the details. I still get chuckles at lab meetings and things where some basic biology technique which you learn in year one biology, I don't know because I've never had a biology course. But that's fine. You have to put your ego aside sometimes to be able to really forge ahead into new areas. So that's kind of what I wanted to let you know, is that you can go from academia to industry, you can go to government, then go into academia, and then switch careers, in terms of, engineering into nanoscience, into I'm now a professor of biomedical sciences in the med school here at UCF, but also a professor of chemistry, physics, biomedical engineering as well. And so for all those, you had to be actually, you voted in by the faculty, so you had to show competency in all those. But it just shows that you can do a lot of different things with your degrees in your experience. So don't hold yourself back as to what you think you can and cannot do. Give it a shot. You may not succeed, but if you don't ever try, you don't ever know. That's all I got to say.
Thanks, Dr. Hickman. Next, we'll welcome back Dr. Morales-Pantoja to speak about working in academia.
Well, there is so much I relate from every panelist that have spoken, and I think I mean, it is clear that for all of us there is this tendency and the need to do something impactful, right? Whether we are in academia or in industry, policy, I think that's the common thread that we have here. And for me, the way I started in academia, my career as a research scientist, it was as an undergrad. I did my undergrad in the University of New Mexico in Albuquerque, and I was really fascinated with biochemistry and the neurobiochemistry. So my research really was based on studying protein aggregation and the antioxidant response in the animal model. So I started with animal models of multiple sclerosis. And this was, I was just amazed and feeling privileged to be able to do research on an area that is close to my heart, which is multiple sclerosis. And while I was doing all of this amazing research, the tipping point for me to change to human models was the fact that all the research I did, while it was informative and of course relevant to a degree, a lot of what I did not translate to human models. So what I did as an animal model for multiple sclerosis, only a small portion of that was able to translate into human samples of multiple sclerosis patients. So I then, when I did my PhD at Hopkins in School of Medicine in cellular and molecular medicine, really my aim was to study directly from human samples. And so I had the opportunity to work with cell lines from different patients of multiple sclerosis and with different degrees of disability. And for me, this was so important because, in neurodegenerative diseases, or pretty much any disease that affects the brain, we have the constraint of accessibility to the tissue. And so working with stem cells directly from human patients opened the door for me to get even more involved and more invested in understanding the mechanisms of neurodegeneration. So I work with these cell lines and and healthy controls, and I recapitulated or mimic inflammatory aspect of multiple sclerosis, trying different cytokines that are relevant to the disease. And so, for instance, I was working, and part of what I was working it was to understand why there is this failure of remyelination in patients with multiple sclerosis, especially when we know that there is a pool of progenitor cells that should be able to regenerate. So for me, it was very important to find that the stem cells are able to differentiate into these oligodendrocyte cells, but it's the inflammatory environment that prevents their regeneration and remyelination of the brain. And so while working with all of these cells, it was a mix between 2D cultures and 3D. Certain phases were 3D, but most of them was 2D. And while working on all of this, I realized that this was great, but we needed to take it to the next step to work with more complex tissues. And as I was working in my PhD, actually, there was this press release from CAP with the brain organoids. And so that was really like, okay, so this is really what I want to go next, to increase my understanding of neurodegeneration in more complex tissues. And so I finished my PhD and transitioned to my postdoctoral research here at CAT. And I started working with brain organoids. And during this transition, also I had the opportunity to, okay, so it's like, okay, I know that this is a great platform, but I also know that we need to improve our models to get it closer to human relevance. And so, you know, part of the research I showed you. And so I then shift my focus slightly into improving the in vitro systems to make them more relevant, because as I mentioned, obviously, if you make them more physiologically relevant, then you have a better platform to study diseases and what goes wrong, right? And so I focus my research in the glia enrichment, now working with the microfluidics, which is this part of what resonates from Dr. Hickman, when it's like, okay, just, you know, try and do different things. So going from, you know, undergrad as focus being biochemistry, neurology, then stem cell neuroscience, in neuroimmunology as my postdoc, now increasing the complexity and transitioning to bioengineering with this microfluidics that we're working here at CAT. And so that's how my career has started and has shifted slightly, going from mechanisms of disease to improving microphysiological systems or in vitro systems to study neurodegeneration. And the idea is to go back and circle into mechanisms of neurodegeneration now that, you know, the field is so really exciting right now with all the microphysiological systems and the improvement of all of these platforms. And so this is really what has guided my career as in academia.